at the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's Science Tonight. Now here's your host, Chris Smith.
Good evening, everybody. Welcome to Science Tonight. Thanks for joining me. I'm your host, Chris Smith. It's great to be with you, as we are every Thursday night at 7 o'clock Eastern, right here with the North Carolina Museum of Natural Sciences. But, of course, you may have noticed things are just a little bit different, like that swanky new intro video and the new name. We used to call this program the Virtual Science Cafe, but I think we're going to keep doing this thing online. We're going to keep live streaming the show every week, so it's got its new name, same place, same time, though. Welcome to Science Tonight. Thanks for joining us. Listen, we've got a great show. I know it's going to be a great show because we have a great guest. We have a great guest who came up with an amazing title for tonight's program, Crabitat. Isn't that great? Don't you love it? Crabitat. Although I went and I actually Googled Crabitat to see if like it was a thing already, just to know. And maybe I was also trying to find the YouTube link for tonight. I should have had that already. But what I found, Erin, this might interest you, is that Crabitat is hermit crab aquariums.
Huh.
They call them Crabitats.
I didn't know that.
So when I looked up Crabitat on YouTube, I just got dozens of videos of people setting up aquariums for hermit crabs. But hermit crabs aren't the topic for tonight. Let me introduce our guest. You've already heard and seen Erin now. Erin Voigt is a PhD candidate at North Carolina State University studying juvenile blue crab and the habitats that they live in. Erin also works for the North Carolina Sea Grant and Pamlico Sound, Pamlico Estuarine, National Estuarine Partnership.
Yep.
APNEP.
Yep, that's it.
I'll drop a link in the chat a little bit later for everybody, which I should say, it's not just going to be a program between us. You all at home, wherever you're watching from, can join in as well. Use the chat box or the comment thread if you're watching on YouTube or Facebook, and drop your experiences with blue crab maybe in the chat. Tell us your stories and ask your questions. A little bit later in the program, I'll be grabbing your questions and posing them to Erin. Sound good? Sound good?
Yeah, sounds great.
All right. Well, Erin, thanks for being here.
Thank you for having me. I think this is the first time I've gotten to get dressed up out of yoga pants or pajamas in 9 months, so I'm very excited to be here.
You look
out of the
wonderful.
house. Thank you.
So let's jump in.
Mm-hmm.
You're a Marylander.
I am
Originally
born and raised. Yep.
And when I think of Maryland, I see the flag, the like black and yellow stripes,
Yep.
and the red and the white.
Yep.
And then right in the middle of the flag is always like a big crab.
Yeah. We are often associated with the crab. In fact, generally, one of the most common names for blue crabs is the Chesapeake blue crab, which obviously relates to the Chesapeake Bay, and I have a little bit of qualms with it because, as we'll see later tonight, I study the same blue crab in Pamlico Sound, so it's not just the Chesapeake. The blue crab actually has a pretty large area, and it can take up many different habitats in the United States, including Pamlico Sound.
Oh, nice, nice. Okay. So, tell you what, before we jump right into your research and what you study,
Okay.
I have a little game prepared.
All right.
I hope you'll indulge me.
Sure.
I'm calling this game System Preferences.
Oh,
To
okay.
gauge your preferences for some of North Carolina's greatest stuff.
Oh, man.
Being from Maryland, but now having spent time in the Carolinas, are you up for it?
Yes.
Okay. First off,
All right.
Pepsi or Coca-Cola?
Coca-Cola.
Ooh, okay.
Although, I'm going to be honest, I don't think I can taste a difference. I know that's like a very, I'm sure the internet's going to be angry at me about that.
You've got the chat box open. Let us know, all right? Each one, let us know your preferences too, each one. All right. Next one. Krispy Kreme or Dunkin?
Ooh. My sister lived in Philadelphia for a while, and I have a lot of friends up there, and I feel like they're going to be really upset, but I like Krispy Kreme.
Oh, okay.
I do. They're better. Sorry.
No, I like that answer.
Okay, good.
I like that answer. Okay, Bojangles or Chick-fil-A?
Chick-fil-A. I know I might get kicked out of the state for that, but I don't like Bojangles that
I
much.
might get kicked out.
I know.
You're like 1 for 3 now. Okay, let's bring it a little bit more close to your research area.
Sure.
Old Bay seafood seasoning. Or
You don't even need to let
Zatarain's
me know.
Oh,
It's always Old Bay. Always Old Bay.
always Old Bay.
Always. There is no other alternative to Old Bay.
Yeah. Okay. That's it. I'm gonna check it.
Old Bay is king.
What are people over here? Okay, you've got lots of Pepsi fans in the chat box right now. Okay, let's see. Just a couple more. Hard shell crab or soft shell?
I am a hard shell person. I gotta be honest with you. Soft shell crabs freak me out a little bit. I think it's something about the legs. I have a rule where I don't eat exoskeletons. It's just all of them. So I prefer hard shelled crabs. Yeah, I like picking them. Gives you a lot of time to have good conversations with friends.
There you go, and you get the great texture and the sound, the crunch.
Yeah, exactly. You get to take out some anger, maybe a little bit of stress from your work week, really smashing through those shells, for sure.
Beaten. All right, last one, and this one ties directly into your science. Chesapeake Bay blue crabs or Pamlico Sound blue
All
crabs?
right. So similar to the Pepsi versus Coke thing, I don't think I can taste the difference. I can taste the difference between like very salty and maybe a more freshwater caught blue crab, but I can't taste the difference between the Chesapeake Bay and the Pamlico. And as we're trying to, it's like choosing between your children, the body of water I grew up on or the body of water I'm getting my dissertation in. There is no favorite.
That's a very safe way to play it.
Thank you. I thought so.
That's very good. That's very good. And somebody watching on YouTube says that Maryland approves of your answers.
Oh, thank you.
So,
That's good. That's good to know.
All right, let's get into it. So,
Sure.
give me the sort of elevator pitch for what your research is about. We said blue crabs and their habitat. So, what's going on in the Pamlico Sound?
All right, yeah, so I actually study juvenile blue crabs. So unfortunately, not the type of blue crabs that you would generally catch in a crab pot. Instead, I study crabs that are only about this big. And in particular, what I'm looking at is I want to know what type of habitat they tend to live in at the highest quantities. So in Pamlico Sound, there are two primary types of habitat. You have either seagrass habitat, and I could actually pull up an image of that right now, just in case you wanted to kind of get a visual of what I'm talking about. So you can either have seagrass habitat, which is a submerged vascular plant that lives underwater, or you can have something called shallow detrital habitat. I generally refer to this as SDH. And basically what SDH is it's this type of habitat that we find a lot in Pamlico Sound, and it occurs directly on the erosional side of a marsh. So you can see it underwater in the picture on the left, or you can actually see it from the air as the brown zigzags in the aerial photo. So these are the two main habitats in Pamlico Sound, and I want to understand which of these habitats is used more by the majority of juvenile blue crabs.
So the crabs have preferences for different types of habitat?
There are some preferences. A lot of it is going to depend because when crabs are really small in their smallest form or one of their smaller forms called megalopa, they're actually slightly planktonic. So what that means is that they can't swim against water currents, and so they get pushed by the water currents and end up kind of stumbling upon habitat, and then they can choose to settle into that habitat or not settle into that habitat. So a lot of what I look at with the blue crabs is going to be not necessarily a choice where I give them two habitats and say, like, which do you like? Which people have done. You can actually do that with juveniles. Instead, what I'm doing is I'm going out into these habitats in Pamlico Sound. I am looking to see what the density of blue crabs in those areas are. I'm also looking to see how large of an area the habitats take up, and then I'm looking at something called the distribution. So whether or not there's a lot of crabs here and only a few crabs here, or whether or not it's like pretty evenly spread out across the entire habitat. I'm looking at those three things in order to figure out where the majority of the crabs are.
So I'm going to back things up a little bit. Let's go back to the beginning, because that was a great 60-second explanation, 1 minute thesis. That was perfect. But so let's dive in a little bit. Get it, water. Nobody at home laughed at that joke. I know you didn't. So if we go back to the beginning for the blue crab, you mentioned that a baby blue crab is called a megalopa.
Yep.
But they're planktonic, so they're not crabs?
So they are. So blue crabs are really neat, and this actually applies to a lot of marine invertebrates altogether. They have something called a complex life cycle. So I can actually show you an image right here that looks at what the blue crab life cycle looks like. And so what happens is, you're probably familiar with the adult blue crabs, which you see off on the left. When they find a date and get together, the female will become a sponge crab. She'll migrate all the way to the inlets. So in Pamlico Sound, that's Hatteras Inlet, Oregon Inlet. And then what happens from those eggs on her stomach is she releases something called a zoea. And a zoea actually looks a bit like an alien. So here's a photo of one taken underneath a microscope, and that thing in the bottom is actually a quarter. So to give you an idea of what size they are.
Oh, wow.
Yeah, so these guys look like aliens. They actually go all the way out into the open ocean and hang out on the continental shelf for about a month before kind of going through another major body change or a metamorphosis into something called a megalopa. So this is a megalopa. You see a very zoomed in image of it on the right. To give you an idea for the size, that little speck on my pointer finger on the left is a megalopa.
Wait, right there at the joint?
Yep, yeah, very,
wow,
I think,
tiny.
Yeah, right there, very small. So they're super, super tiny, and these guys actually get blown back into the estuary, and they're gonna be the ones that are kind of looking for that good habitat. And then once they find it, what happens is they'll go through one more body shape change. When they find the habitat, they say, great, this is where I want to live. And then they're going to settle and turn into the type of crabs I study, which are what we call instar crabs. So here's a photo of them. These guys look a lot more like a blue crab. They basically are miniature blue crabs, but as you can see on the photo on the left of it on my finger, they're still pretty small.
That is a baby blue crab.
Yes, very tiny. They can be as large or as small as about 2 millimeters, which is about the height of a penny if two were stacked on top of each other.
I'm very surprised at the way that the first two larval stages?
Yeah.
That we would say, okay, the first two larval stages look nothing like.
Yeah, I think most people would not guess that that was a blue crab if I had shown that to them without any sort of context.
You ever go up to people and you're like, guess this animal? And they completely get...
It's mostly, I have it and I'm like, did you know this is a blue crab? So that's, yeah.
When you can go to parties.
Yeah, exactly. That
thing.
is my party trick.
That's your I love it. I love it. That's great. So here, looking at the crab life cycle, we saw different places where different crabs are hanging out at different points in their life. So they're using this great variety of shoreline. Who goes where?
Okay, so that's actually a really interesting question because, as I mentioned before, right, so blue crabs, because they are planktonic, they're really getting pushed by oceans and currents and wind currents. So there's a lot of factors that decide where a blue crab kind of goes. And in particular, there are two mechanisms that we think of when we think about blue crab transportation, especially at the juvenile stages. The first happens in the absence of hurricanes. So I've got a little bit of an animation just so you can actually see the blue crabs moving. That might be a little helpful to the people at home. Great. So what you can see is you see those seagrass beds along the eastern shore of Pamlico Sound. So those are all those little green bushy things towards the east. And on the western shore of the sound, you see the SDH habitat. So that's that marsh adjacent habitat. So when there aren't any major hurricanes or tropical systems coming through the area, the blue crab, the megalopa, will get pushed in through the inlets and will settle and turn into instars right in those seagrass beds. So they kind of use the seagrass beds as a type of like landing zone. But as you can imagine, as more and more blue crabs come into the system, it starts to get a little crowded in those seagrass beds. And then they'll go through something called density-dependent secondary recruitment, which is basically just a really large, like, scientific name for them hopping up, leaving that seagrass habitat, and then moving across the entire estuary to settle into that SDH habitat.
Moving to the suburbs.
Yeah, basically, exactly. But there is another major transportation pathway that can happen. Blue crabs recruit into the estuary, or they come back into the estuary from the open ocean in the late summer, early fall. So as anyone that's lived in North Carolina knows, that is the same time as peak hurricane season. So the other major transportation mechanism of blue crabs is going to be storm driven. So when you have these large hurricane systems off our coast, they actually can blow the megalopa through the inlets and past those seagrass beds, and they will directly settle into the SDH habitats. So those are your two major mechanisms, and it's really important because it shows the different ways that crabs get to both of those habitats, both the seagrass and the SDH. And that's a big part of my research, is understanding how these transportation pathways lead to different levels of abundances in both habitats.
So I have a sense that we know a lot about seagrass beds.
Yes.
Lots of things use seagrass, and so do the blue crab. Do we know as much about the shallow detrital habitats, or is that why we have Erin Voigt?
Yeah, no, that's a huge part of my dissertation research. So the seagrass habitats are mapped about every 4 years. In fact, APNEP, part of the groups that fund my research, they do a lot of this seagrass mapping. So I have these great aerial photographs and GIS documents that show me exactly where and how much area the seagrass takes up. However, if you remember when I was kind of first introducing my dissertation research to you, I said that in order for me to really understand how blue crabs use SDH for seagrass, I need to know three things. I need to know the density, I need to know the area, and I need to know the how well blue crabs are dispersed in the habitat. So I know a lot of that for seagrass, but for SDH, I don't. We have no idea what causes it or where you can find it. So a big part of my research has actually been going out and sampling to see where we do and do not find SDH. Furthermore, we're also looking to better understand the factors like the wave force, or possibly what type of marsh species lives on the fringe, the fringing shoreline. So we're looking at all of these different factors to try and come up with a model that will allow us to predict whether or not SDH is there or not.
So if you find SDH, you know where you've got some. How do you find those teeny tiny blue crabs?
That's a great question, and we use a couple different methods. So I think a lot of people, when I tell them that I study blue crabs, they get pretty excited because they think I'm going out and I'm using crab pots, and I'm going to not only, catch stuff for research, but, maybe after research, a little bit of dinner. Wow. Unfortunately, that's not quite how it works. Crab pots are one of the only sampling things that I do not use in my research. Instead, what I do is something called suction sampling, which is a lot like an underwater vacuum. So I've actually got a photo, and I'll walk you through this photo a little bit. But here is one of the technicians in my lab. Her name's Aurora, and she's suction sampling. So the first thing you'll see is this large red ring. There are actually 2 of them in this. And these are sampling rings. And it's what we use in order to standardize the area that we are sampling to find blue crabs. If you remember, I think I used the word density. So blue crab density is the number of blue crabs per meter squared. So it's based on the area. So we take
red floating ring.
Yep, a red floating ring. So it's basically the bottom is weighted and it's got a metal ring on the bottom to hold a perfect area. And then it's basically a cylinder of mesh with floats at the top. So it almost looks like a kiddie pool if you cut the bottom off of it. So we put that out, and then if you look kind of closely at this, you'll see that Aurora is holding what looks to be a PVC piece that's kind of shaped like a T, and on the one side it'll have a bag on it. And so what we do is we are able to suck up water, and in doing so, we suck up baby blue crabs and we push them into that bag. And then over on the boat, by me, there is a big suction pump, and that's what's causing the suction, and then it flows through that tube right there. So yeah, that is the suction sampling apparatus, and that's how we sample the majority of blue crabs in Pamlico Sound.
So what's the data say? SDH compared to not SDH, compared to those eastern seagrass beds? What are you finding?
So what we found was actually pretty interesting to us, and it was really unexpected. So what we generally think of in Pamlico Sound is that you have the SDH along the eastern shore, and then you have SDH along the western shore. And so the crabs are kind of making this choice not only in terms of the habitat, but also spatially. However, what we found is there are actually some really tiny ephemeral seagrass beds. So they're really small, the seagrass is fairly patchy, and some years they're there and some years they aren't. So you can't always really track or map them the way you would the seagrass along the inlets on the backside of OBX or the Outer Banks. But what we found when we collected blue crabs in those habitats, so the eastern inlet adjacent seagrass, which is really thick and lush and is always there, the western SDH and the western ephemeral seagrass, what we found is that that ephemeral, that really patchy seagrass on the western shore, it actually has somewhere between 2 to 3 times the amount of juvenile blue crabs than the other 2 habitats. So that was really shocking to us. These itsy bitsy tiny habitats that don't take up very much area at all are just crawling with juvenile blue crabs. So that was a big, exciting finding for us.
The western seagrass beds got the new hip coffee shop, and
yeah, exactly.
Everybody moved.
They're the cool neighborhood.
That's the cool neighborhood. So what does this mean then for people who like to eat blue crab?
Yeah, so
it's a huge fishery in
it is,
right?
So it's the reason that we do this research. I mean, obviously it's important for us to understand as much as we can about blue crabs, just from a scientific perspective. However, I like to do applied research that kind of feeds back into the community as well. And for me, the reason that I got into this is because, as most people around the coast know, the blue crab prices have gotten pretty expensive, and that a lot of that has to do with the fact that the blue crab standing stock is not great right now. So I actually have a graph that looks at what we call the standing stock biomass of blue crabs. And standing stock biomass is just a really complicated word that basically refers to the reproductive population of blue crabs. And so as you can see, it was pretty steady from the 1985-ish through 95, 96. Then you see this huge spike in the abundances of blue crab, which is wonderful. But then immediately following that, around 2000 or 1999 to 2000, you see that the blue crab population drops off pretty remarkably. And in fact, if you look at that red line, so that is the mean or the average standing stock biomass, you'll see that after 2000, we only crossed that line once in 2003. But otherwise, we've remained under the average amount of standing stock. And this has led DMF and other fisheries agencies to consider the North Carolina blue crab a concern for overfishing. So obviously, as someone that loves to eat blue crabs, likes blue crabs generally as an animal, I want to investigate why this is and why that blue crab population hasn't come back. Because with an overfishing event, we restricted fishing pressure on them, but we never really saw a comeback of blue crabs. So one thing that both DMF as well as several researchers that look at blue crab abundance and population in North Carolina have kind of decided on is that we think that it has something to do with the recruits or the amount of juveniles coming back into the system. So one of the things that I'm very curious about is I want to be able to understand which of these habitats is the most important for conserving in order to bolster those juvenile blue crabs so that they can replenish the system. Because as you know, everyone's a little strapped for cash, but conservation agencies especially are. So while we know that blue crabs use seagrass and SDH, generally there isn't enough funds to go around to protect a whole bunch of different habitats all over the place. So what's really important is that we know where the major areas are that need protecting, where the majority of the juveniles that mature into the adult population, where they're coming from. That way conservation and restoration agencies can really target their money towards those areas and kind of be the most efficient, get the most bang for your buck essentially with blue crabs.
That's incredible.
Thank you.
I mean, like, yeah, that's amazing. I mean, to situate the work that you're doing into such an important context, right, of that resources are scarce, especially for conservation efforts. And so let's solve the problem. Let's figure out how and where we can do the most and the best work. That's
Thank
really
you.
interesting.
That's what I'm aiming for.
That's what you're aiming for. I love it. That's great stuff. So, last question that I'll throw your way, and then I'm going to kick it to the folks who are watching online. So everybody, get your typing fingers going and drop some more questions into the chat box to take us through to the end of the show. I've seen videos online and GIFs, whatever people want to call them, of crabs with knives.
Yes, that's terrifying.
and I don't know how the crabs got the knives, but people keep their distance from a crab with a knife.
Yes.
What can you tell us about angry crabs?
So people keep their distance from a crab normally, especially a blue crab. Blue crabs and maybe stone crabs, but I personally think blue crabs are the angriest. So as much as I talk about blue crabs in terms of their fishery role, so how much humans eat them, one of their major ecological roles, so like the role they play in nature, is also that they're just really tasty. So a lot of recreational fish that people enjoy catching on the weekend, like red drum, love eating blue crabs. It's their favorite food as well. So blue crabs get eaten a lot. And as a kind of to deal with that, they tend to be a bit grouchy. Honestly, they're a bit aggressive. So if you've ever had the joy of coming into contact with a blue crab, you will know the general blue crab, I want to fight you stance, which is, you know, arms out, pinchers ready. Exactly. Ready to meet you. And what's really interesting is that while a lot of people are familiar with the adult blue crabs doing that, even these tiny, like 2 millimeter blue crabs that I catch absolutely want to fight me every time. Like that is their go-to, you know, my finger against their tiny little claws. But interesting, if you ever wanted to give a blue crab a knife, which I'm not sure why you would want to do because it would be very scary. But the way you can do it is because they pick up their arms and put out their claws like that when they feel threatened, you can pretty much hand them whatever you want. So when we're doing research on the larger adult blue crabs and we don't want to get pinched, we'll often put a pencil in front of their face because they'll grab onto that and not us. So yeah, you can pretty much hand a blue crab whatever you would like to.
The pencil is mightier than the sword.
Yes, it is. Although not mightier than a blue crab, we have lost many a pencil to a blue crab snapping it in half.
Oh my goodness. And the blue crab, do they know to go for like that fleshy bit right in the middle between the fingers?
Scientists have not proven that, but personally, I think so. Like, I think they know where weakness is and they go for it. Yeah, absolutely.
They're smart fighters.
They are.
I love that a tiny little 2 millimeter crab is just like, come at me.
Yeah,
Right.
absolutely. All of them. Every time. I think even in the megalopa stages, they would like to fight you. Yep.
Ready to go. Aaron, this has been amazing.
Thank you.
Thanks
It's
for
been
sharing so
great
much
to
of your
be
work.
here.
Okay, I'm going to go now to the chat box. Everybody, let's see what you're thinking about and see if we can't get some answers to your questions. First off, okay, let's see if I can actually read this one. Bronwyn Williams, I'll warn you, Bronwyn Williams is the research curator for non-molluscan invertebrates
Oh, okay.
with the North Carolina Museum of Natural Sciences. I'm going to out you on your questions, Bronwyn. Do you consider blue crab to include all of the species in the genus Calanectes found along the mid-Atlantic coast, or just Calanectes sapitis?
That's actually a great question. And to kind of catch some of the viewers at home that might not be as familiar with the taxonomy of swimming crabs in Pamlico Sound. So Calanectes sapitis is the blue crab. It is the actual species of blue crab. However, there are a number of other species, including one called Calanectes similis, which is named strictly because it looks very, very similar to the blue crab. In fact, one of the only ways you can really ID the difference is to do a lot of kind of complex taxonomy on them. Once they're adults, you can count the number of ridges between their eyes. They also tend to be a bit smaller. But for my research, and this is actually a great question, in the fisheries world, a fisherman will take a Calanectes sapitis, the blue crab, or Calanectes similis, as long as it's of the right size, and we'll take that in as a blue crab. So we've chosen, because this is a fisheries kind of focused study, that we count both similis and sapitis in our general groups right now. However, I don't know if you saw in one of my previous slides, one of the ways that we sort all of these blue crabs is via image processing. And that actually allows me to zoom in close enough that I can start to differentiate between sapitis and similis. So for right now, my numbers do combine them, but we have the ability to differentiate down the line if we would like to.
All right. And yeah, Bronwyn had that follow-up, which I think you answered. If they were lumped together, how does it affect the interpretation? But it does make sense if, with the fisheries focus, you want this to be useful to fishermen and to the fisheries scientists as well. So if that's the way they're counting them.
So, and I will say, like, a lot of so my study is building off 2 prior grad students who have also done blue crab research in Pamlico Sound. And both of them did separate out similis versus sapitis. And so I think for my final results, I will most likely go through all of these images and ID them out properly before going to publication. But for right now, we are grouping them together.
Excellent stuff. A series of really good questions here. I'm going to start with this one. What time of year does your research take place, and how does that coincide with the crab's habitat usage?
That's also a great question. So I mentioned it really, really briefly that my research takes place during what we call like blue crab recruitment period. And the blue crab recruitment period generally occurs between August and October. And as, like I said before, if you're familiar with the North Carolina weather cycles, you know that's peak hurricane season. And so another thing that you might be familiar with, especially if you live on the coast, is that our winds actually shift during that period too. During the summer, the winds primarily come out of the southeastern area. And then in the in the fall, going into early winter, we start to get nor'easters, which are wind patterns coming from the northeast. And so that switch from the southern winds to those nor'easter winds are actually what blows the megalopa into the sound. So yeah, this period between August and October is really, really important, and it's when I sample all of my blue crabs.
How has your research been impacted during hurricane season?
That is also a great question. So unfortunately, let's see, I've sampled for 3 years, 2017, 2018, and 2019. And we have not been able to fully sample in September for 2 of those years because we had a series of tropical storms in 2017 that were causing small craft advisories for about a week and a half there when I was trying to sample. And then in September of 2018, we actually had to be evacuated out of my field sites because of Hurricane Florence.
Oh, wow.
Yeah. We tried to get out there and sample before it came through, and we got a couple of sites done, but not everything. So yeah, it's definitely makes things very interesting logistically and planning wise. However, I always like to point out that hurricanes are actually pretty good for blue crabs. It's a little bit of like a Goldilocks scenario. So if they're too strong, they can have some really dire effects, or if they drop a lot of fresh water and rain, it can be pretty, have a negative effect on the blue crab stock. However, tropical storms, especially those that kind of just skim the coast or come right in, are actually really wonderful for blue crab recruitment. And in fact, that year where you see that really high spike in blue crab standing stock, the reason that we think that we see such a high rate of blue crab abundance during that year is due to tropical storms kind of blowing a lot of the megalopa into the system during that time period. So hurricanes can actually be pretty beneficial to the crab, although fairly difficult for me.
I just picture a blue crab, there's a hurricane coming, there's an adult blue crab, claws out, like waiting. Bring it on. Okay.
Yeah, exactly. I feel like their aggressive nature really, yeah, they're into hurricanes.
Born in them and molded by them.
Yeah, exactly.
All right. Now, you talked about the mapping, and we saw some of the aerial imagery of the SDH, shallow detrital habitats. So this is a good question. How has the use of drone or mapping technology contributed to your research?
Yeah.
They ask what other tools, but we talked about suction sampling and being out on the boats.
Yeah, for sure. So
about drones and mapping.
I have a little bit, well, we'll talk about it first. But so one of the major things that I had kind of pointed out earlier, and I haven't talked too much about the results of that because we're still processing a lot of this, but I need to understand where SDH is and what causes it to occur. So we know that SDH is essentially the part of a marsh. Here, I will pull up the SDH photos so you guys can all kind of picture what I'm talking about a little bit more. So SDH is basically the part of the marsh where there used to be vegetation. The vegetation has died off, and you now have this big mat underneath that's full of all of the roots and shoots of the dead marsh vegetation, and blue crabs love it, so it sinks under the water. So we know that erosion, in particular, this loss of the vegetation line, is really important to forming SDH. But while there's a lot of really great research looking to better understand what causes marsh erosion, there are so many factors that can affect it that it's often a little bit difficult to predict on its own. So one of the chapters of my dissertation is using drones to take these really high resolution maps of these SDH areas and non-SDH areas. And what I do with those is I create a map that I'm then able to measure the shoreline and look at how it changes over time. So similar to the image on the right here, this is a single shot of one of those. And basically I'll take a whole bunch of shots like this, and we'll stitch them together like a quilt in order to make a giant map. And then I'll trace it over, and I will do that approximately every 3 to 4 months. And I'll be able to basically create this really high resolution change in shoreline, which then I can compare to in the field cores that I've taken. And I can say, okay, well, we know that the shoreline is changing at this rate, and we know that SDH is here, whereas we know the shoreline over there was changing at that rate, and we know that there was an SDH there. So it gives us this ability to basically build a model that will predict where you see SDH and where you don't.
And then where the baby blue crabs are going to be.
Yes, exactly. And so that kind of builds into this whole stacked analysis of knowing, you know, the density of the blue crabs plus the area of the habitat. Exactly.
Excellent, excellent stuff. All right, so I think we've got a Marylander in the chat box here. They want to know why Chesapeake crabs are so much larger.
Are they larger?
So they said the largest blue crabs I've seen in North Carolina are the size of small or medium ones in Maryland. There are no large, extra large, or jumbos here.
Oh, I'm not sure there's data to support that.
Okay.
I believe our crabs are about the same size. I would have to look up the actual landings, and if you really want to get into the data of it, you can actually go on to North Carolina DMF's website, and they have all of the size distribution of the adults there. So you could physically compare whether or not Maryland and North Carolina blue crabs are different sizes. I have bought crabs from both places, and I don't think, I mean, I've bought jumbos in North Carolina, so they do exist here. Maybe it's who they're getting their crabs from.
It's that preference for a Chesapeake Bay crab.
Yeah, I think
maybe
they just look bigger, just knowing. Maybe it's the Old Bay. It is. Once you put Old Bay on a crab, it just looks bigger.
It just looks bigger.
It's tastier, and it looks bigger, yeah.
Okay. We've got some time still left in the show, so let me grab some more of these questions. Let's see. Ephemeral seagrass, are there less predators?
That is a great question. So I am probably not going to be able to do it with my dissertation, but one of the next steps, I think, to my research would be to better understand both the food sources in these three habitats, as well as to understand like what the predation rates are in these three habitats. My guess would probably be not particularly, and here's why. So believe it or not, one of the main predators of juvenile blue crabs is adult blue crabs. Blue crabs are incredibly cannibalistic. However, I can say from being in all of those habitats, there are plenty of blue crabs in all three of them. So there are a ton of blue crabs in the eastern seagrass, there's a ton of blue crabs in the ephemeral seagrass, and there are a ton of blue crabs, large adult blue crabs, in SDH as well. So in terms of the predator amount, I don't think that's what's driving it. My guess for why you see such high densities in the western seagrass beds is that if you kind of picture what I was talking about before with the transportation mechanisms, the blue crabs will come in and they will settle directly next to the inlets. And we've actually done some studies where we look at the distribution of blue crabs in eastern seagrass beds and western seagrass beds. And what we find is that blue crabs are mostly just found around those inlets. So I've got a map out that might make this a little easier. I know I'm kind of going off topic for this question.
No, let's see.
I think it's an important side note to talk about. So this is a map, and what you see in green is that eastern seagrass bed. And at this level of kind of zoom or aerial You can't actually see the western seagrass beds. They're so small that you can't see them when I have a map this large. Instead, I had to zoom in on Stumpy Point, and you can see that kind of in that call-out window over to the left. And there's this itsy-bitsy tiny sliver of this western seagrass bed. So what we know is when blue crabs come in, they tend to come in through Oregon, which is up here, and then Hatteras Inlet, which is down here. And they'll settle a lot in these seagrass beds that are just adjacent to those inlets. However, all of the seagrass that you see like up in what I consider like the elbow of Cape Hatteras, so over by like, not Salvo, but over in the elbow of Hatteras, you don't tend to see as many blue crabs over there. So it's really, there's kind of this gradient where as you get farther away from the inlets, you start to see fewer and fewer fewer and fewer blue crabs. However, when you switch it over and you look in the western areas, so both the SDH and the seagrass over there, what you find is that it doesn't vary that much. So if you sample down east in Hyde County, kind of over in this area, versus if you sample up here, the amount of blue crabs are about the same. And so what we think is happening is that they kind of fan out. So they come in through those inlets, they're super concentrated right at the inlets, and then they fan out across the whole western habitat area. And so because they are able to kind of have access to a greater amount of habitats, they're going to look for those like really prize habitats. And for them, that's these seagrass beds. So I think the reason that we see such high densities of blue crabs is because they can kind of choose what habitat they want to live in there. And so when they can find that really good seagrass, that's what they choose to settle in.
All right. Excellent stuff. All right. There's just a few more here.
Okay.
Glenn wants to know, is the decrease in blue crab numbers related to water quality?
So possibly.
Do we even know?
So I kind of hop into that question a step down the line. So the first question would be like, so why would water quality affect blue crabs? Blue crabs, I say this with the greatest amount of love for this animal in my heart, are a bit like cockroaches. They can live anywhere. In marine labs, it's not uncommon to find blue crabs living in the drainage system. I have also found blue crabs in like these marsh puddles that have like slime mold growing on them, and the blue crab is as happy as can be. Where we think that water quality might come into play is with the habitat, in particularly seagrass habitats. So my research wants to look at, have we seen these habitats changing over time? What do we know about which ones they use? Could it be that there may be an issue where these nursery habitats that are so crucial for the blue crabs are actually being hurt by water quality or are changing and decreasing over time? So that's kind of where I come into it. It's a step down the line, but it will get at beginning to answer that question.
Melinda's asking, what is the weirdest tool you've used in the field?
What is the weirdest tool that I've used in the field?
I mean, the red floating rings, that's just...
The floating ring is weird. The suction sampler is just a strange thing. Trying to think, I've always...
giant underwater vacuum?
Yeah, it's literally a homemade underwater vacuum. That's exactly what it is. I've definitely used some weird... Oh, here's one. I recently had to buy a food dehydrator, not to make fun, yummy snacks, although I do have one of those at home. Instead, I had to buy it because the seagrass that the blue crabs live in is really, really thin and small. And in order for me to kind of measure how much there is, I need to dry it out. And the drying oven I had on campus would get so hot that it would actually like bake the seagrass and burn it. So we needed to get something that would dry the seagrass out without baking it. And so what I ended up having to buy was just like a normal cheap food dehydrator from Walmart. And that's what I've been using. Yeah.
Can you bag it and sell it as kelp in a health food store?
Maybe. Yeah, seagrass is not tasty. I've tried. It's not great.
You've tried. Not great. Okay, let's see. Olivia Coretti is asking, is SDH found anywhere other than Pamlico Sound? So could your research be applied to other estuaries?
So this is actually a really cool question. Thank you, Olivia. Because one of the things that we're trying to do with my research is to come up with a quantitative definition of what SDH is. So like I said before, there have been a number of grad students before me that have researched this, and kind of the way that we knew what SDH was is that we go to the site where the SDH is, or our advisor, Dr. Dave Eggleston, kind of shows us those sites, and we look at the ground and we go, that is SDH. And that's how we've known what it is. We described it with words, but we've never described it with numbers. So
you know what it is, you know what it is.
Yeah, exactly. So what I've been doing is I've been taking a number of cores and areas that have been sampled since the 90s as known SDH locations. And then I take a lot of samples in areas that I know are not SDH. So either sand patches, or if you're familiar with the term like pluff mud, which is that like really silty mud, or just other marsh adjacent habitat that I know not to be SDH. And so we're taking a number of soil characteristics in order to come up with this quantitative definition for what SDH is and what SDH is not. And one of the main uses of this information, not only for my own study, but is that I can publish this and other researchers and other places can take a sediment core, process it the same way we did, and can say, okay, this is SDH. This is what they've been talking about and citing in their papers for so many years. We do know that it's here as well. And my guess is, yes, absolutely. In places like the Chesapeake Bay or any sort of estuary that tends to be microtidal in particular, you are going to most likely find SDH there.
All right, excellent stuff. And we got a little bit more on your research in there too.
Yeah, a little bit.
The work you've been doing. All right, it looks like our last question. I don't know what this is. Maybe you'll know what this is.
Okay.
A couple of people have mentioned it now. Have you played Fight Crab?
No. So I'm pretty sure I don't know exactly how to play Fight Crab.
Is when you fight a crab?
No, I have played that game, and I have lost that game a lot. Like a lot. No, I'm pretty sure Fight Crab is where you take two crabs and you put them in a confined space and they fight. I don't know if someone in the chat can confirm whether or not that is what Fight Crab is. I haven't played it intentionally. I've definitely played it probably accidentally a couple times. For example, when we take samples, we generally, I vacuum up all those blue crabs and we put them in a mesh bag. We have to open that up and remove any large blue crabs from there, because if we leave the large blue crabs in there, they will eat the rest of the babies in there. So we have to take them out, measure them, and throw them into the water. Because if we keep them in the bag with all of the juveniles, I will be missing some juveniles when I get back to the lab. So I guess that's kind of like Fight Crab, I believe, if I know my internet references.
Yeah, I might have to.
I might have to Google that one. That might be a YouTube for after the show tonight.
I just assumed that it was a video game.
Oh, is it?
I went with there's just a crab and you
And maybe, is there an app? If there's an app, I would like to get that.
Crabs.
Yeah, I mean, I personally have tried to fight some blue crabs. It never goes well for me. I always end up bleeding. So.
Well, I'll wish you better luck than against the crabs in the future.
Well, I study juveniles. They can't hurt me.
They're this big.
Yeah, exactly.
Well, Erin, this has been fabulous.
Thank you.
Thank you for being the first guest on the new Science Tonight.
It's been a privilege to be here and to chat with you. I also wanted to do a quick shout out. You have already talked a lot about them, but science is not made on an island. It really takes a village. So I've got a quick slide real fast just to throw up and to thank all of the different people that have helped along the way, the Eggleston Lab, all of the technicians, as well as all of my funding sources, Southeast Climate Adaptation Science Center, North Carolina Sea Grant, and AppNet. Awesome, yeah.
Absolutely.
And thank you guys for all chiming in on this wonderful Thursday and asking such great questions. It's been great to get out of the house and chat about blue crabs. Some of my favorite things.
Fabulous stuff. And I see so many names on your thank you slide. Great partners of us here at the North Carolina Museum of Natural Sciences, too. So I would also like to thank them for supporting
yeah, and thank you, Chris.
Research.
Thank you for having me.
Oh,
giving me an excuse to do this.
This has been fun. I've learned more about blue crab than I think I ever would have ever.
Awesome.
And now people can come back and watch this video later, and they can learn about blue crabs too.
Sweet. I'm excited.
And then when your dissertation's done, we'll have you back here at the museum, and you can wrap up the very end of that story with a neat.
Absolutely. Yeah. I'll have an actual answer for you guys. Which habitat is the more important or the more valued? Pending. Trailer.
There we go. Hey, everybody. Thanks for tuning in to Science Tonight. I hope that you enjoyed the program. Remember, you can visit naturalsciences.org and check out more virtual events that are going to be happening from now until wherever. I hope that you'll come back for the next edition of Science Tonight as well. We'll be back here not next Thursday. That's Thanksgiving, so happy Thanksgiving, everybody. We won't be here then, but the week after that, the first Thursday in December, we'll be here with the author of Wicked Plants, Amy Stewart. So that's going to be a great conversation. Don't miss it. Go ahead and mark your calendars, set your bookmarks, or hey, you can just subscribe to the museum's YouTube channel right there below this video and click the little bell icon to get notified, and then you'll get the notification to come and join us for the next edition of Science Tonight. You can also get more information about what's going on here at the Museum of Natural Sciences by following the museum on Facebook, Twitter, and Instagram. We're at Natural Sciences on all three platforms. Great education, great learning, great pictures and videos. You will enjoy following the Museum of Natural Sciences on social media. I promise it. Until next time, everybody, take care, stay safe out there, and have a happy Thanksgiving. We'll see you next time. Bye, everybody.